Hi students, welcome back. So this video we are going to continue with the May-June 2022 5-2 that we started, the planning and the analysis paper of summer of 22. So in my previous video, I had discussed about the question 1, and we had started with question 1, part A. Part B, we had finished this big planning question, which is usually for 7 marks, and we had discussed about the shortcuts here. Now we move on to the second part of the B question. As I mentioned in all my videos, and I keep reiterating that please make sure you mark the things which you have to do. So it says here, calculate the percentage change in volume of the red tissue in each cube. State how the students could calculate the percentage change in volume of the red tissue. So whenever we are talking about percentage change, in this case, there are no values which are given to us. So everything that matters for 2 marks is the formula which you are going to write. So the formula for any type of percentage is usually the initial volume. But because we are talking about this specifically, we have to write the initial volume of the red tissue in each cube minus the final volume of the red tissue in each cube. Please make sure you write off the red tissue, because specifically they have mentioned in the question, they have mentioned it twice in fact. So you should be utilizing it, divided by the initial volume of the red tissue in each of the cubes, and then make sure you multiply it by a factor of 100, because that's how you're going to get a percentage value. So this question is for 2 marks. Why is this for 2 marks? Because the first part, that is this part, gets you 1 mark. Giving the multiply by 100 gets you the other mark. So please make sure that although this looks very simple, but any question which comes for percentage change in temperature, volume, any specific topic, please make sure that you write the formula. If you have the values also, 1 mark is always for writing the formula which has to be there. So with this, we are done with the second part. Now we move on to the next question of the first question itself. The next question, which is the part C. So the part C usually varies from being an experimental question where you have to interpret a particular graph. Sometimes it is about certain calculations which have to be done. Sometimes it is about descriptive questions which can be there. So please make sure that, as I have said, you read through it, skim through the question, or read through it a little bit faster. So the first one comes in a further experiment, the student investigated the effect of temperature on the release of pigments. So before you are starting, please have a picture. You've already answered a few questions in reference to the experiment earlier. So the earlier experiment was actually talking about how there was a change in volume. Okay. How there was a change in volume with respect to the surface area volume ratio. Now we have investigated in this experiment the effect of temperature on the release of these pigments also. First, we were just doing surface area volume ratio. Now we are looking at temperature itself. Cubes were cut, beakers containing water were prepared, beakers were placed in water bath, beakers were allowed to equilibrate for 5 minutes, beetroot cubes were added to beakers. After 30 minutes, beetroots were removed. Colorimeter, each with blue-green filter, were used to measure the percentage transmission of light through the fluid remaining in each beaker. At this point of time, when you're reading these points, it might be that only 2 or 3 is something which you understand, because our main aim right now is to go to the question part. And as you can see, there is actually a table which is showing up at the bottom, and that's what we are going to move. So here, there is a table which is right here, and it is showing the temperature and percentage transmission for light for 4 different students, and they have taken the mean. So obviously, if you see in this question, you see this particular part empty. So we know that a question is soon going to come about this empty part where they might ask us to calculate the mean. So as we move further, we are expecting that question is going to be there, because the place is empty. The student decided that 2 of their results are anomalous. One of the results that they consider anomalous is circled in table 1.1. Circle the other anomalous result and complete the table by calculating the mean percentage. Now here it is again a 2 marker. So you're looking at something which is an anomaly, or something which is not following a particular pattern. The word anomalous means something which is not normal, or something which is not following the pattern. So let's look at our first set. So as the temperature increases, we can see this is 100, 83, 82, 26, 1, which is right there. And as the temperature is increasing, there was a decrease right here. Now, again, we see kind of a pattern which is right here. Now, again, so what are we looking at here? Do we look at the columns or do we look at the rows? So first, we start looking at the rows, because how will we say that the result is anomalous in any one of them? It has to compare with something. I should say that this value is normal because it was normal in the other 2 experiments. So we are going to compare 100 with 97, 90 and 96, which is comparable. 83 with 92, 96, 86, kind of comparable here. 83, 86 are comparable. So we do a tick. In 45, we already have it, so we can ignore this line. Then we come to 65. We have 26, 31, 29, 17. Okay, so this is a line we'll be looking at, or the other one. Now at 85, we have around the same range. The missing is just about 1 number. The result which looks anomalous, if you look in the line of 65, we have 220's value and 31, which is quite near. However, this value of 17 is quite far away. So now the question had asked us for an anomalous result, and how do we mark it? So please make sure that you use either a dark pencil or you use a pen to circle 17 properly. Now, the next part which comes in is to calculate the mean. How do we calculate the mean? Make sure you show your working. It is in that column. So mean is equal to addition of all the values divided by the number of values. So we have 4 values here and we get an answer. Now, whatever answer you get has to be with only 1 point. You can see the answer comes out to be 89.3 if you do it on the calculator. But if you see after the point, there is only 1 digit. So it has to be there. Even if let's say in this case, the answer was coming out to be 89, you have to make sure that you write it as 89.0. Even if it is coming out to be a whole number, the way you are going to write the answer really depends on how you are actually going to put it and analyze how answers are written in that column. So please make sure you don't make that mistake, because if you're going to make that mistake, you're going to lose that 1 mark, irrespective of the fact that, you know, you've done your calculations correctly, and you might end up losing the mark. It is a 2 marker, very easy to score. Now comes the next question. So the C second part comes in. Describe any two ways in which the method which was described in C has been improved to give better repeatability between the results of different students. Now they are saying that how do you think this has been changed so that it describes two ways in which this method could have been improved. So here you have to think of what all can be used. So let's look at the first one. It says cubes of beetroot were cut. Now here they've not mentioned about the size of the cubes which were cut. So here we can mention about, we can use the point of size of cubes should be kept constant, or we can say that the age of the cubes or the beetroot which is used is kept constant. The second point says beakers containing water were prepared. The beakers were placed in water bath. Now, here the water is prepared, that is okay. Now, the beakers were put in water bath at the temperature between 0 to 85 degrees Celsius. That is also correct. The beakers were allowed to equilibrate for 5 minutes. So the beakers were allowed to adjust for about 5 minutes. The beetroots were then added to the beakers. Now, when we add the beetroots here, did we make sure that the beetroots were covered with water? Did all of them have the same volume of water? So when we are talking about water here, we can think of while we are putting, is it the same volume of water that we put? Did we make sure that our beetroots were covered or not? After 30 minutes, the beetroots were removed. Okay. Now we move to colorimeters. It says colorimeters, each with the blue-green filter, were used. So here they are saying that they are using multiple colorimeters, which can lead to multiple errors. And we don't want any errors to happen at this point of time. So we should be using one colorimeter. Okay. One colorimeter that is there. Also, as I had mentioned in my previous papers, also whenever you are using the word water bath or colorimeter, please try and use the term digital. So either you say thermostatically controlled or digital water bath or digital colorimeters is what you are going to be talking about. We are in a century where we are using electronic items. We are not using something which has been designed manually. We are not heating up the water and putting in water bath. So please make sure you do not make your mistake. So coming back to our question. So when you were approaching that question, as I had said, make sure you go to the answer above. So we've actually got a lot of points where we talked about. Coming back, let's have what all did we talk about. We said we can have the size of the beetroot, which can be maintained. Okay, that is one of the points. The second point is that we have to make sure that all the colorimeters are the same. Okay, the colorimeters are the same. We've already got our 2 points, but we mentioned that we can have the age also, which can be same. So there is more point here. We have to make sure that our beetroots were covered in water. Our water bath was thermostatically or digitally controlled. So we have a good amount of points. In this type of a question where they've asked you to make a change according to something which was given to you, always go back to that and talk to yourself while you're attempting each of the points. Okay. So let's move on to the next question. As we move on to the next question. The last part of question number 1, it says with reference to table 1.1. Now, what is table 1.1? This was table 1.1 where we took out the mean and we looked at a pattern. State and explain. So here again, 1 mark is for stating, 1 mark is for explaining the effect that increasing temperature has on the release of beta-lane. Now, what all does it have as an effect? Let's go and see. As we increase the temperature, what happens? So as we increase the temperature in this table, if you look very carefully, the percentage transmission of light kind of decreases. So when we are saying the percentage transmission of light decreases, that means there is more of pigment. Because if there's less of pigment, there will be less reflection of light. So please think very carefully. More light will mean a lighter color. So the statement which comes in here is that as there is increase in temperature, as there is increase in temperature, okay, as there is increase in temperature, our pigment released. There is more red color which is there. That's why absorbance decreases. So we have more beta-lane pigment that is released. Please make sure you write the statement properly because that is 1 mark. That is your state mark. Now we talk about the explain part. Now we talk about the explain part. Why do you think that is happening? As we are increasing the temperature, what all can happen due to high temperatures? If we go back to our study, permeability increases. Okay, permeability increases. Also, when we are talking about high temperatures, we can talk about the word of denature. Maybe a membrane protein has completely denatured. So even if you're using that word, it helps us a lot. Increase in permeability, increase in fluidity if you want to talk about with increase of temperature, or you can use the word denature, that the membrane is kind of denatured at higher temperature, so it's damaged. So here the key word which you're going to use is denature. That's your explanation. So it will be very easy for you to score that 1 mark because you just have to write the statement, but make sure you go to the explanation. Talk to yourself, how do I explain it? It can denature. That's what we are talking about. So now we move on to question number 2. So question number 2, as I had mentioned earlier, question number 2 is kind of related to mostly inheritance or the coefficients or the chi-square tests or the null hypothesis that we talk about. So here, if you skim down, there is question on immunity. And as we go down and down, there's variable, standardized, and we can see a question on null hypothesis right here. And then there's a question on the graph and a little bit of explanation. So it is a 12 marker. It is not that difficult to score in paper 5-2. So when you are giving your AL exam, paper 5-2 is the one which is a scoring paper. So as we come, we are talking about the European badger. And as I said, always read through the points in this paper. Even though you do not understand them in the first go, it's still perfectly fine. It is still perfectly fine that you don't get it. You can always come back like we did it for the previous one. You have the paper with you. Badgers can contract bovine tuberculosis caused by a particular pathogen. Researcher estimated that 5% of the population was infected with this bovine tuberculosis. It spreads between badger population and other population of wild and farmed animals. Then we have herd of dairy cows produce milk. If dairy herd becomes infected, the pathogens can be transmitted to the humans. A variety of measures have been introduced in an attempt to reduce the rate of this bovine tuberculosis and so prevent infection of dairy cows. These measures include vaccination and removal of badger. population. Excuse me. So now we've already marked onto the important points which were there. So we can go on to our questions. It's about TB, we know, and something which is being transmitted. Now we come to a 3 marker. So at this point of time, we are not going to get any question more than 4 marks. It'll be 3 or 4 marks because our big question of 7 or 8 marks is already come in the first question. So the second question subparts will be of relatively smaller points. Suggest how researcher estimated the percent of badger population that was infected with bovine tuberculosis. So they are asking you, how did the researcher estimate the population that were affected by this tuberculosis? So basically, they are asking you a way that in a particular population, we have the population having a particular disease. How are we going to estimate the percentage? So here we are going to a chapter of selection and variation where they are asking us to find the percentage of our badgers, a percentage of an individual. So how do we do? First of all, we take a big sample. That is one of the most important things. So we take a big or a large sample. Whenever you want to estimate, you have to take your bigger sample. And in that sample, you have to find the number. So you find the number of the ones which are normal and the ones which have the BTB. Okay. Now, how do we know which one had the BTB? Did we do a test? No, we did not mention that we did a test. So now, after we have counted all the individuals, we have to make sure we do the test for BTB. The test for BTB was done, and we have to find out which one of them are mostly positive. Okay. Now, which one of them is positive? And the fourth one, how do we calculate the percentage? How do we calculate the percentage is that we take the number which has the number of the population of the infected individual divided by our total population multiplied by 100, and we will. So you have to write that this is how we are going to calculate. And what method do we use? So if we go back to our studies from the book, we had actually used something which is called as the Lincoln Index. So the Lincoln Index is usually used in this process. Let's say you forget to mention this point. It's fine. It's fine even if you've forgotten about it, but please make sure, even if you've forgotten about it, that you write these steps in the right manner. It is very important to write all of these steps, even though they look like 4 and we will think that we need 3. But the entire process is supposed to follow a procedure. And we need to make sure that we have followed this procedure when we are answering the question. So I'm repeating the question again. It's asking you, how are you planning to get the sample? First, you take the big sample, then you count all the individual, then you test them, and then you calculate the percentage. So I hope all the students have written about it, and then we move to the next one. And as we move to the next question, we have the A part here and it doesn't have any subparts. So the next part is going to be the B part. Now we have, is it possible that the wild badgers, if infect the dairy cows, it is possible that they infect the cows with BTB. The group of scientists investigated the correlation between BTB in cows as well as the badgers, and there were 30 study areas. So here they are doing approximation random selection. The number of infected cows and number of infected areas were counted. the number of infected badgers were determined, and the number of infected cows was determined by testing for response. So here again, I said the reading has to be a little bit faster here. Even if you're not able to understand each of the line right here, it's perfectly fine. Just read through it, that what are they trying to do, and try and understand. So here they are trying to compare the affected badgers affecting the cows. So if you look at the table, talk to yourself, area where counts were made, A, B, C, D, blah, blah, blah. They've given all the areas. Year when the counts were made, they mentioned the years. Now we come to our question. The scientists expressed their data on numbers of infected cows and infected badgers at the population density of infected cows and infected badgers. Now, obviously, we don't have any data yet. So the question is going to be on this line, which has been just written above, on this line. So it says, explain what is meant by the term population density and suggest why the data is expressed in this way. So first, we have to give the definition, or we have to explain what is meant by population density. So population density was a term which we had studied when we were studying the population and the counting chapter. It's basically the number of individuals. Okay, and here we are talking about the badgers. So number of individuals per unit area. If you do not remember it, please note it down. Make sure you write it on a sticky note. Stick it where it is visible to you every day. These are the terms you need to remember. Population density is number of individuals per unit area. And suggest why the data was expressed in this way. Why do you think they are asking? So here you've read the question and you're still confused that I have to suggest the way. For what way? Talk to yourself again. As I've been saying, the scientists express their data on the above line, if you read, on numbers of infected cows and infected badgers as a density. That is, they are using per unit area. So we are using it in areas rather than picking one-one comparison, because we allow comparisons here. The reason behind it is we are allowing comparison between a number of them. That is, between the infected cows as well as the one which are the infected badgers. So we are allowing comparison. Any population density experiment which has been done allows for comparison. So here, first was writing about population density. What does it mean? And then why was it being done? So that we can make comparisons. Now they're asking you, state 3 variables that were not standardized during this investigation. So 3 things which they have not mentioned about in this investigation, that was not standardized, that is not fixed, or they've not done any testing. So what all has been studied? First thing is what all has been studied. So as I said, when you're answering this type of question, make sure you go back to your data. So it says 30 study areas in England were identified. So obviously the number of study areas which was there was standardized. So there's no problem here. Each of the areas was approximately 100 kilometers per hour. 100 kilometer per square. So this is also standardized. 10 out of 30 study areas were randomly selected. Okay, this is also, they are doing random selections, so there's no problem. The number of infected cows and number of infected badgers were counted in each area. That is correct. So they've standardized this. But did they standardize the cows? Okay, since we are talking about cows here, did they standardize the cows? So did they standardize the type of breed or anything which is talked about? No. So the type or the breed of the cows or the badgers is not something which has been estimated. Okay. Now, we move to the next point. The number of infected badgers were determined by testing for present in tissue sample taken from the badgers. So we've just picked up one point on the top. Now they're talking about the fact that they have been doing the testing. But by what method? Was the method standardized? No. So we need to know by what method was it done. So there is no mention about standardization of the method. The number of infected cows was determined by testing for response to introduction of antigenic material into the skin. So they are saying that they have done this test by introducing an antigen material into the skin, but they have still not mentioned here that it was standardized on certain samples or not. Each badger might react in a different way. Each cow might react in a different way, which is not known. So we've come up with 2 points, that is the type and the method, which was not standardized. But if we look at the table, the year is also not standardized. They have not mentioned anything about the year. They've not mentioned about the season. Maybe the season is the winter season where most of the badgers don't move outside. So doing the testing, they're very inactive at this point of time might be one of the reasons. So any season or time of year has not been standardized. So we already have 4 points here. So let's go back to our question. We already have the 4 points right here for our question. It says state 3 variables which were not standardized. And when we are talking about these 3 variables, we talked about the type of the cows which were used. We've not type of the breed. That has not been standardized. A method was used, but there is no mention of method being standardized. They did not mention anything about the year, nothing about the season. So here you can pick up any of the 2 points. You can create a lot of them. This is the same process we followed for the previous setup of questions we had, when you have to go back and reread again. So that's why your first read should be faster. But this read, when you're answering the questions, has to be very clear so that you can scrutinize each point so that you can write the answer. And now we come to a null hypothesis question. The scientists decided to analyze their data on the occurrence of the bovine tuberculosis in cows and badgers. State the null hypothesis for this. So we are trying to establish a relationship between infected badgers and infected cows. So what will be our null hypothesis? That there is no correlation. So null hypothesis always starts with no correlation between the infected. Please make sure you use the word infected, the infected cows and the infected badgers. So either when we are talking about null hypothesis, we have to use the word correlation. Please make sure that the word correlation is made for null hypothesis. Do not forget that term. Now we have a big scatter graph, which is given in the second part. And there will be a table which is given right below it, where they have mentioned the areas and the relation. So here they've already moving on to a null hypothesis. They've found that the relation is positive, and in 2 they found it negative. Significant, non-significant is what the data has been given. Now we come to the last question, where it says that some of the scientists suggested that the information showed a strong positive relationship between the number of infected badgers in an area and number of infected cows. These scientists also suggested that the bovine tuberculosis could have been reduced by removing the badgers through culling. Evaluate whether or not data in this table and the figures support the suggestion made by the scientists. Now we have to look at the graph as well as the table. Now, if you look at the graph, okay, so here they're asking in the question whether or not the data supports or not. So here this is asking you to write in support and in not support. So first part which you will start is that you have to write in support. Okay. And we cannot write unless and until we have a look at both of them. That is the table as well as the graph. So if you look very carefully here, we have the graph which is given to us, the scatter plot, and we see that the number of infected cows per kilometer square. There are a lot of dots on the top part, and there are 1, 2, 3, 4, 5 dots on the bottom. So if we look at the support data, what do we say? That yes, it shows in most of the areas, that is our data set 1 and 2, in all the sampling areas, the first one, the combined data which is there shows a positive correlation. So yes, it is supporting what is shown in the graph. Okay. This is our statement for whether it supports or not. Whether it supports or not. So what do we pick up that will be a support here? So first point we mentioned about the positive correlation. Also, when we are talking about positive relationship, we can also use the word we just mentioned in null hypothesis, that it shows a significant correlation. So whenever you are answering a question based on null hypothesis, please make sure you refer to the table. Two of your terms are right there. One is it is showing combined data showing positive correlation and positive relation and significant correlation. So we can use these 2 points as the 2 points which are right there. Now we come to something which is not supporting. So here we can mention either from A to H, they show negative correlation in the table. And if we go back to the graph, it shows that A to H, which are there, show negative correlation, and it is not very significant. This is when we are talking about the table, right? Now, if we have to write the same from the graph, because they've said we have to talk about the graph too. So when we are talking about the graph, we will have to do a correlation or a number or a data which is given. So if we look very carefully, we don't find any data between 0.4 and 0.6. So if we look, there is no data between 0.4 and 0.6, which might raise questions. So that is one of the points which we can use. We find data in all data points, but we have these one set of data points between 0.4 and 0.6 that it has happened. So we are there with our 3 points. Coming back to how to write the answer now. First part is, first and the second part we were able to pick up for support. Okay, we were able to pick up that there was a positive correlation and there was a significant correlation. So positive from the table and significant from the table. Now, what does not support? What does not support from the graph? Okay, what does not support from the graph is that A to H, you can see, are all together in the graph, whereas D to J are all together in the graph. And similarly, we find the same in the table, that they have shown a negative correlation and a non-significant conclusion. So please make sure you write from the table, use the term from the table. So we are done with the paper 5. Please post your comments. Let me know if you have any questions or any problems, and we can address those while you're answering these questions. Please make sure you read the question first. As I said, there are certain precautions when you're attempting the paper 5. First, read fast and then read slowly when you're attempting the questions and when you go back. Okay, see you. Take care and all the best for your exams.